time for the test. So I'm going to read it aloud to you like I normally do, and then encourage you to follow along. And if you have an IP or a 504 where it's suggested that you have tests read to you, definitely take advantage of this. But here we go. I'll read the question, and remember, you answer. You can always blow up the size of this on your Chromebook. Let's begin. Question 1. In the diagram below, what is the length of the garden snail from the beginning of its foot at point A to the end of its foot at point B? Your answer choices are A, 2.6 millimeters, B, 26 centimeters, C, 26 millimeters, or D, 260 centimeters. Question. Bell just rang, but that was yesterday, not now. Question 2. Which part of a compound light microscope should a student adjust to allow more light to pass through the specimen? A, diaphragm, B, fine adjustment, C, stage, D, ocular. Question number 3. A slide of human blood cells was observed in focus under the low power objective of a compound light microscope. The microscope had clean lenses. When the microscope was switched to high power, the image was dark and fuzzy. Which parts of the microscope should be used to correct this situation? A, the nose piece and the coarse adjustment. B, the diaphragm and the ocular. C, the diaphragm and the fine adjustment. D, the objective and fine adjustment. The illustration below represents photographs of cells viewed through a compound light microscope. Photograph 1, photograph 2. What should a student manipulate to change from the view shown in photograph 1 to that of shown in photograph 2? A, the nose piece, B, the fine adjustment, C, the eyepiece, D, the stage clips. Question number 5. The diagram below represents a microscope field of view that has a diameter of 2.5 millimeters. A protist is shown in the microscope field below. Which action would center the specimen in the field of view? A, move the slide 2.5 millimeters to the left towards A. B, move the slide 1 millimeter to the right towards B. C, move the slide 2.5 millimeters to the right towards B. D, move the slide 1 millimeter to the left towards A. Question number 6. The diagram below represents a single layer of cells seen in low power, 110x, in the 110x field of view of this microscope. If kept in this layer, what is the total number of cells that would be viewed in the high power or 400 visual field of this microscope? A, 1, B, 2, C, 8, D, 32. Oh, I love this question. The diagrams below show 4 different one-celled organisms, shaded, in the field of view at the same microscope but at different magnifications. Which illustration shows the largest one-celled organism? A, which is at 400x, B, which is at 100x, C, which is at 400x, D, which is at 100x. Question 8, a student prepared a wet mount of the letter E from a newspaper. He then placed a slide on a stage of a compound light microscope and adjusted the stage clips as shown below. Which drawing most closely represents the image of the letter E that would be seen by the student when using the low power objective? A, B, C, or D? The diameter of a field of vision of a compound light microscope is 1.5 millimeters. How would the field of vision be expressed in micrometers, or as I call them, ohms? A, 150 ohms. B, 1,500 ohms. C, 15 ohms. D, 15,000 ohms. Should be on question 10. A protist. Question 10. A protist measures 500 micrometers in diameter. What is the diameter of this organism in millimeters? 500 ohms. Is it A, 50 millimeters, B, 500 millimeters, C, 5.0 millimeters, or D, 0.5 millimeters? All right, I need to take a break from reading. All right, here we're at part 2 of our test. Sorry, I had to take a little break while reading the test. I had to use the lavatory, as you can imagine. Okay, let's see what we got here. No, not that one. Where is it? Where is it? Where is it? There we are. Okay, there it was. All right. Should be on question 11. Wasn't that a nice little break there? A student took a swab of cells from inside of her cheek and transferred the cells onto a slide. Under a microscope, the diameter of the field of view shown was 1.25 millimeters. What is the approximate diameter of each individual cheek cell? A, 150 ohms. B, 50 ohms. C, 250 ohms. D, 350 ohms. Question 12. The diagram below shows cells as seen in the high power 400 field of view of a compound light microscope. What's the approximate diameter of cell A in micrometers or ohms? Is it A, 100 ohms, B, 1 ohm, C, 250 ohms, D, 150 ohms? Ooh, lucky number, question 13. To locate a specimen on a prepared slide with a compound microscope, a student should begin with the low power objective rather than the high power objective because the field of vision is larger under low power than it is under high power. B, because the portion of the specimen that can be observed under low power is less than the portion that can be observed under high power. C, field of vision is smaller under low power than it is under high power. D, the specimen does not need to be stained for observation under low power, but must be stained for observation under high power. Question 14. The diagrams below represent wet mount microscope slides of fresh potato tissue. A drop of stain is put in contact with the left edge of the cover slip on slide B, and a piece of absorbent paper is placed in contact with the right edge of the cover slip. What is the purpose of this procedure? A, it helps increase the osmotic pressure of the solution. B, it prevents the stain from getting on the ocular of the microscope. C, it prevents the water on the slide from penetrating into the potato tissue. D, it allows the stain to penetrate the potato tissue without the removal of the cover slip. "15. The diagram below shows how a cover slip should be lowered onto some single-celled organisms during the preparation of a wet mount. Why is this the preferred procedure? A, because the possibility of breaking the cover slip is reduced. B, the possibility of trapping air bubbles is reduced. C, the organisms will be more evenly distributed. Or D, the cover slip will prevent the slide from breaking. "16. What activity might lead to the damage of the microscope and a specimen? A, focusing with low power first before moving into high power. B, using the course adjustment to focus the specimen while under high power. C, cleaning the ocular and objectives with lens paper. D, adjusting the diaphragm to obtain more light under high power. Question 17. The diagram below represents two cells next to a metric measuring device under the low power objective of a compound light microscope. What is the approximate length of a nucleus of one of these cells? A, 1,000 ohms. B, 500 ohms. C, 100 ohms. D, 1,500 ohms. Question 18. According to the cell theory, which statement is correct? A, cells are basically unlike in structure. B, cells come from pre-existing cells. C, viruses are true cells. D, mitochondria are only found in plant cells. Question 19. The cell theory states that A, all cells regenerate and contain the same basic structures. B, living organisms are composed of cells that arise from pre-existing cells. C, organisms that lack certain organelles reproduce by binary fission. D, all cells have nuclei that contain genetic information. Question 20. What cell structure makes a plant cell more rigid than an animal cell? A, cell membrane. B, cytoplasm. C, ribosome. D, cell wall. Question 21. Your lungs provide the site of gas exchange for your body. What organelle serves the same purpose in an animal cell? A, ribosome. B, mitochondria. C, cell wall. D, cell membrane. Question number 22. Which part of the cell allows nutrients and other materials to enter or leave the cell? A, nucleus. B, chloroplast. C, cell membrane. D, cytoplasm. Question number 23. Man, this time flies when you're having fun, doesn't it? Question number 23. Molecule X. Molecule X moves across the cell membrane by diffusion. Right? We have a chart here with 1, 2, 3 columns and 4 rows. Right? Which row in the chart above best indicates the relationship between the relative concentration of molecule X and the use of ATP for diffusion? Is it A, row 1, high concentration moves to low concentration, ATP is used? B, row 2, high concentration goes to low concentration, but ATP is not used? Row 3, low concentration moves to high concentration and ATP is used? Or row D, low concentration moves to high concentration, ATP not used? Question 24. Molecules A and B come in contact with the cell membrane of the same cell. Molecule A passes through the membrane readily, but molecule B does not. Which one of the following statements could describe molecules A and B? A, molecule A is a simple sugar and molecule B is a starch. B, molecule A is a protein and molecule B is a fat. C, molecule A is a starch and molecule B is a simple sugar. D, molecule A is an amino acid and molecule B is a simple sugar. Question number 25. I feel like we've seen this question before. It says the diagram below shows the relative concentration of molecules inside and outside of a cell. So the circle is the cell, and the key says the diamonds are protein, oxygen is squares, glucose is stars, and carbon dioxide are the ovals. Which one of the following statements best describes the general direction of diffusion across the cell membrane of this cell? A. Carbon dioxide would diffuse out of this cell. B. Protein would diffuse out of this cell. C. Oxygen would diffuse into this cell. D. Glucose would diffuse into this cell. Question number 26. Structures adapted to convert stimuli into nerve impulses are known as A. Muscles, B. Effectors, C. Receptors, D. Glands. Question 27. A hawk sees a field mouse, which it captures for food. What do the eyes of the hawk function as in this activity? A. Stimuli. B. Neurotransmitters. D. Receptors. C. Receptors. D. Effectors. Let me read those answer choices again because I feel like I stumbled through them. A. Stimuli. B. Neurotransmitters. C. Receptors. D. Effectors. Question 28. The secretion of chemicals that stimulate responses in specific tissues is a function of A. The nervous system only. B. Neither the nervous system nor the endocrine system. C. Both the nervous system and the endocrine system. D. The endocrine system only. In humans, question 29, in humans, one difference between the nervous and the endocrine systems is that A. Nerve responses are of longer duration than endocrine responses. B. Only the nervous system plays a role in homeostasis. C. Only the endocrine system secretes chemicals. D. Nerve responses are more rapid than the endocrine responses. Question 30. The graph below shows the levels of glucose and insulin in the blood of a human over a period of time. This graph represents A. Autotrophic nutrition, B. An antigen-antibody reaction, C. An allergic reaction, D. Maintenance of homeostasis. Question 31. What usually results when an organism fails to maintain homeostasis? A. The organism becomes ill or may die. B. A constant sugar supply for the cells is produced. C. Growth rates within organs become equal. D. The water balance in the tissues of the organism stabilizes. Question 32. If frog eggs taken from a freshwater pond are placed into a saltwater aquarium, what will most likely happen? A. Water will neither enter nor leave the eggs. B. Water will leave the eggs. C. The eggs will burst. D. Salt will leave the eggs. Question 33. A wet mount of red onion cells, as seen with a compound light microscope, is shown below. Which of one of the following diagrams best illustrates how that technique would most likely be used to add salt, or to best illustrate the technique that would most likely be used to add salt to the red onion cells as shown? Is it technique A, technique B, technique C, or technique D? Question 34. This is the final question on our test today. You guys have been awesome. Give yourself a pat on the back. You're so awesome. And remember, if you don't like the way you're doing this test, take it again. Take it 100 times. Take it 1,000 times. You got until the end of the quarter to get it done. All right? Why not? Number 34. The diagram illustrates what happens when a particular solution is added to a wet mount slide containing red onion cells being observed using a compound light microscope. To observe cells on the slide shown, it is best to start out using the A, high power objective and focus using the fine adjustment only. A, low power objective and focus using the fine adjustment only. B, low power objective and focus using the coarse adjustment. C, high power objective and focus using the fine adjustment. D, high power objective and focus using the coarse adjustment only. And that's it. Woo. Remember, send me a message if you want to redo it. You can comment in the classroom, and they'll send me an email, and then I'll reset that for you. All right. And if you're done with the test, remember the only other thing that's due today is the microscope lab 1 and 2. Microscope 1's past due, but microscope 2 is due actually today. So that's what I would use your time, especially if you have lab. You guys are awesome. Stay gold, Pony Boy.